Hello all, in this video we will learn about the transgenic animals. Transgenic animals are the animals with the modified genome. A foreign gene would always be inserted into the genome of the animal to alter its DNA. This method is done to improve the genetic traits of the targeted animal. The improvement of genetic traits was done by selecting the breeding methods. The animals with desired genetic characteristics will be mated to produce an individual with improved genetic characteristics. This technique is usually time consuming and expensive. So later it was replaced by the recombinant DNA technology. Transgenesis is the phenomena in which a foreign gene with certain characteristics will be inserted artificially into the genome of a targeted animal. The foreign gene that is inserted is known as the transgene. The animal whose genome is altered is known as the transgenic animal. These genes are passed on to the successive generations. The transgenic animals are genetically engineered and also known as genetically modified organisms. The first genetically modified organism was engineered in the year 1980. There are certain methods for creating the transgenic animals. The first one is the physical transfection. In this method, the gene of interest is directly injected into the pronucleus of the fertilized ovum. It is the very first method that proved to be effective in mammals. This method was applicable to a wide variety of species. Other methods of physical transfection includes particle bombardment, ultrasound, and electroporation. The second method is the chemical transfection. One of the chemical methods of gene transfection includes transformation. In this method, the targeted DNA is taken up in the presence of calcium phosphate. The DNA and calcium phosphate co-precipitates which facilitates the DNA uptake. The mammalian cells possess the ability to take up the foreign DNA from the culture medium. Retrovirus. The next method is retrovirus mediated gene transfer. To increase the chances of expression, the gene is transferred by means of a vector. Since retroviruses have the ability to infect the host cell, they are used as vectors to transfect the gene of interest into the targeted genome. The next method of developing a transgenic animal is the viral vectors. Viruses are used to transfect rDNA into the animal cell. The viruses possess the ability to infect the host cell, express well, and also to replicate efficiently. Back to fection. This is the process by which the gene of interest is transferred into the target gene with the help of bacteria. There are certain examples of transgenic animals. The very first one is the Dolly sheep. Dolly, the sheep, was the first mammal to be cloned from an adult cell. In this, in these cells, research for the manufacture of medicines to treat diseases are also going on. The first transgenic cow, Rosie, produced milk containing human protein. This milk contains the human gene and it could be given and the milk could be given to babies as an alternative to natural cow milk. Transgenic animals are also used as model organisms for testing the safety of vaccines before they are injected into the humans. This was conventionally done on monkeys. I hope you would have found the video to be useful. See you in the next video. Until then, it's bye from Radhika.